slide. I just showing you the overall story about this presentation. So the model we used was mouse myocardial ischemia or myocardial infarction induced by ligation of coronary artery for 30 minutes, then remove the ligation and induce reperfusion. So here, this is the left ventricular injury here. And when that happens, the injured cells in this part and leukocytes that migrate into the injury site will upregulate and release cytokines. One, for example, is IL-6, interleukin-6. And cytokines released into circulation and through various mechanisms get to the liver, and the liver will respond to myocardial infarction. One way the liver response is to mobilize or liberate or release physically hepatocytes, liver cells. And the liver cells are liberated into circulation, and a fraction of the liver cells will get to the heart, get to the broken heart here, right here, and settle in the heart or hold to the heart, the lesion of the heart. And what the liver cells do in the broken heart is, and they upregulate and release some protective proteins. These are secreted proteins, small proteins released to the extracellular space. And these protective proteins will act on various signaling pathways in the injured myocardium, and eventually will reduce cell death and protect ischemic myocardium from death or injury. So with that, next I would like to show you where the story started. So when myocardial ischemia is induced like this, this is a mouse heart, real heart. And this piece here is the infarct. And one liver responds to myocardial infarction. And this is how liver cells are physically mobilized. You see, how to read this image here. This was from a liver histological section. The green color is a transgenically induced YFP, yellow colored protein, specifically expressed in liver cells by using transgenic modulation method. And this part, the dark area here, is called central vein. So the liver consists of millions of working modules or lobules, and each module contains a central vein. And these individual cells, there are 2 here and there are 3 there. These are just released from the liver parenchyma or tissue into the circulation. So the released liver cells will get into circulation. This is a blood sample, and you collect blood sample and a smear sample on a glass slide, and you can see leukocytes, this guy here, blue color is cell nucleus here, and green color is liver cells, because only liver cells express the yellow fluorescent protein. Now, when liver cells got into blood circulation, so this cartoon shows the path of the liver cells, where the liver cells go. So here, this is the liver. So this green block is a single liver cell, and this liver cells released into the vein here, immediately will dump into vena cava, the largest vein in mammals. So liver cells can survive the blood flow in vena cava and immediately move to the right heart, right ventricle, right atrium, right ventricle. The liver cells can survive the right ventricular flow, and they get into pulmonary artery right here and go through the pulmonary circulation. So liver cells are happy in the pulmonary circulation, they can survive. And then next move to the left atrium right here and get into left ventricle. So if you get blood samples from left ventricle, you can still see a lot of yellow colored liver cells. So remind you that in the whole body of the mouse, only liver cells express yellow fluorescence protein. The yellow fluorescent protein expression was driven by albumin promoter. Albumin is a protein expressed in liver only, well, mostly. Some other cells may also express, but to a much lesser level. So the liver cells will go with the arterial blood flow, getting into aorta right here, and a small fraction will get into coronary artery right here, into the heart. So that's where liver cells in the broken heart came from. So one thing I like to say is that once liver cells get into the arterial system, they immediately, within a short time, they just disintegrated because of arterial shear flow. And our arterial circulation and the shear stress is very large. So liver cells can, mobilized liver cells cannot just stand the arterial shear stress. They just break down very rapidly. So how you tell that? So you can take blood samples from everywhere, like from vena cava and right ventricle, left ventricle, and from a peripheral artery. If you, for example, take blood sample from femoral artery, so you will not see any liver cells. So in myocardial infarction, by the way, only myocardial infarction induces liver cell mobilization. In any healthy animals or any controlled sham-operated animals, you don't see liver cells in circulation. So a small fraction of liver cells will end up in a broken heart. So this is a histological section from the lesion of the heart. So right here, the left portion right here is the core area of the myocardial infarct. And right here on the right side, there are a few bundles here. Those bundles are surviving cardiomyocytes. And these brown dots are liver cells. So this is using immunohistochemistry to label liver cells by using anti-yellow fluorescent protein antibody. So you see each dot represents a liver cell because only liver cells express the yellow colored protein. So any cells in the heart did not express such protein. So this is the entire path for liver cell, the fate of liver cells after mobilized from liver. Next thing the question we asked was what the liver cells are doing in the heart. So they are mobilized to the heart, they must do something. So another question is how the liver cells are mobilized. So in this presentation I would like just to address these two questions. First, we will start with how liver cells are mobilized. Why myocardial infarction induces liver cell mobilization? As I just briefly mentioned that IL-6, interleukin-6 expression might be playing a role for liver cell mobilization. So this is where we started. And the first thing you do is test if IL-6 is upregulated. So we collected liver heart specimen from the injury site and using immunoprecipitation and immunoblotting analysis to test the IL-6 expression. At different times, 3, 5, up to 30 days, we collected specimens from the heart and just assayed the expression of interleukin-6 protein. You can see that at 3, 5 days post myocardial infarction, you can see a significant increase in IL-6 level in the heart. Similarly, you can test the IL-6 level in the blood in circulation, like using ELISA and so on. Actually, IL-6 level was also upregulated in blood. Next thing we did was try to find out what IL-6 did in myocardial infarction. What we found was IL-6 activates leukocytes. When IL-6 appears, was dumped into circulation and acts on leukocytes in circulatory system. And activated leukocytes will go to the liver. Sure, a fraction will go to the liver and are retained within the liver. So in healthy animals, this is a liver specimen. The green color is a yellow fluorescent protein because only hepatocytes or liver cells express this protein. And the blue dots are cell nuclei. You can see in a healthy animal, or here is a sham operated animal, meaning you cut open the chest wall and you do something on the heart, manipulate the coronary artery, but you do not induce occlusion of the coronary artery. So in this case, you only see liver cells, and sure, somewhere you will see blood vessels and bile ducts. But in my animal mice with myocardial infarction, in addition to the liver cells, green colored, you will also see the red colored leukocytes. So leukocytes were labeled with an antibody against a leukocyte marker. It's called leukocyte common antigen. It's a CD45. So the red color just indicates these are leukocytes. So that means that in myocardial infarction, leukocytes will migrate through the liver capillary wall or endothelial cells and get into the parenchyma of the liver. But when you knock out IL-6, just completely genetically remove IL-6 in the mouse, and you don't see leukocytes retention within the liver post myocardial infarction. But if you inject recombinant mouse IL-6 protein into the circulation of IL-6 knockout mice, and leukocytes retention will appear. So this will suggest that interleukin-6 activates leukocytes, and leukocytes are retained within the liver post myocardial infarction. The next thing is we will ask what the leukocytes do in the liver. So they retain there and they must do something. And one thing actually found that the leukocytes do is upregulate MMP2, a proteinase, a collagenase. So the target of MMP2 is collagen, and also basal lamina and some other extracellular matrix. So these are images from RT-PCR, reverse transcriptase PCR, testing messenger RNA expression of MMP2. You can see in mice with sham operation without myocardial infarction, you only see a minimal basal level expression of MMP2 from liver specimen. However, in MI myocardial infarction, you can see upregulation of MMP2 messenger RNA at 3, 5, 10 days. So this shows that myocardial infarction induces upregulation of leukocyte messenger RNA of MMP2. And next thing we did was to test the MMP2 activity by using demography. You just mix some gelatin, broken collagen molecules in gel block to run electrophoresis gel and see if the liver specimens or lysate will degrade gelatin. If gelatin is degraded, you will see some white band. So in sham-operated mice, you see only minimal basal level of activity of MMP2, or expression level is very low. However, in myocardial infarction, at 3, 5, 10, 20, or 15 days, 20 days even, you can see a significant increase in MMP2 level, meaning that while in myocardial infarction, when leukocytes are retained within liver, leukocytes will upregulate and release MMP2. So that's what this slide tells us about. So now it's liver cell mobilization again, but with a larger magnification. Images showing you the details here. So what MMP2 does is to just break down collagen anywhere here. Say there are many red dots here. Each red dot is a leukocyte, like here. This is a enlarged image from this plot here. So here you see these two liver cells were just mobilized, and there you find at least 1 or 2 leukocytes. So the red thing here is leukocyte CD45, and anywhere you find leukocyte here, like here, there, these red block, the leukocyte will locally release collagenase, the enzyme. Collagenase will break down locally collagen and basal lamina right here in the capillary. Then when collagen and basal lamina are broken, degraded, and leukocyte liver cells just released, mobilized. So here, these three here, a cluster of liver cells, you can also see a leukocyte there. So it seems that anywhere you have a retained leukocyte within the liver, and the liver cells in the neighborhood will be physically mobilized because release of MMP2. So next thing is, this slide shows the appearance of liver cells within circulation. This panel shows the liver cells directly isolated from liver. You just inject collagenase into liver and mechanically dissociate liver cells. And you would like to show you that there are 2 cells mostly you will see in liver specimens. One, these green colored cells, these are hepatocytes, which takes about 85% of the liver cell population. And this green cell with the red circle here, this is a epithelial cells from bile duct. They're called bioductular epithelial cells, the tube that conduct bile from liver. And this slide was from a blood sample. You can see that in a blood sample from the vena cava. Remember that only you get blood samples from vena cava, right ventricle and left ventricle. You can see liver cells. If you get blood samples from arterial peripheral artery, you will not see these liver cells because they just break up. And these look like hepatocytes. The green color, it's a yellow fluorescent protein again. So this is also blood sample. You can see 3 leukocytes. These blue color, the cell dots are cell nuclei. And this guy is a bioductular cell from the liver. Green color shows a yellow fluorescence protein, and red color shows an epithelial cell marker for bile duct. That's a CK cytokeratin 19. This CK19 is expressed in epithelial cells, but mostly in the liver bioductular cells. In blood sample at about after myocardial infarction, you will see about 1.8% of liver cells in vena cava blood sample. That's 1.8% is the peak right at 5 days after myocardial infarction. But after 5 days, the circulating liver cell population just dropped. So this is just a cytometry analysis showing you blood, the population of liver cells in blood. I will just skip that because of time. Next is if liver cells home to or go to the broken heart or the lesion of the heart. This panel shows a section from broken heart. This is myocardial infarction core, and this is peripheral area. You can see a few surviving cardiomyocytes. So this red block is right here. This is magnified. The brown-colored pieces are liver cells. This is used in immunohistochemical assay to label yellow fluorescence protein, because only liver cells express yellow fluorescence protein. Cardiac cells did not express that. In the heart, right here in this area, we found about 4% of liver-derived cells in a broken heart. That's the maximum population. And after at about 5 days post myocardial infarction, after 5 days, the population or density liver cells dropped. At this time, we just don't know what happened to the liver cells, why they disappeared. They might be dead, or they might transform into other cells, but we have not got a chance to look at that issue at this time. So next thing is we asked what the liver cells are doing in a broken heart. So one thing is to upregulate and release cardioprotective factors or proteins. These proteins just reduce the level of myocardial injury or death. The approach we used was DNA microarray analysis. We just screened the whole mouse genome from the liver, specimens from liver post-myocardial infarction at 1, 5, 10, up to 30 days. What we found was there were about 600 genes in liver cells upregulated in response to myocardial infarction. About 300 liver genes were downregulated in response to myocardial infarction. But we did not care much about the intracellular molecules. What we did care was the secreted protein genes, because only these secreted protein genes might be involved in myocardial protection, because the proteins are released and get a chance to get to the heart. So we found 9 secreted protein genes that were upregulated. These include AGP2, that's alpha-1 acid glycoprotein 2, BMP-ER, bumper, that's BMP, bone morphogenetic protein binding endothelial cell regulator. And CXCL13, that's chemokine 13, FGF21, fibroblast growth factor 21, NRG4, neuroregulin 4, and PRG4, proteoglycan 4, and SAA1, serum amyloid A1 and A2, and PFF3, that's triple factor 3. So these are the 9 secreted protein genes that were upregulated in the liver in response to myocardial infarction. And you can see that the maximum level among these 9 genes was about 80-fold increase, 80 times the increase in messenger RNA expression level. That's quite high. And minimal level is bumper here is about 6-fold increase. That's quite high, quite a level of increase. So in terms of time, you can say for most protein genes here, secreted protein genes. The peak was at about 1 day, like 1 day, and some at about half day, like FGF21. Others at 10 days. So different times reflect different functions. Some might be involved in cardio protection in early time, others for myocardial regeneration in later time, such as TFF3. So what are the functions of these proteins? So far, none of these proteins was identified as myocardial protective proteins. So what we know about the functions of this protein from literature is AGP2 is an inflammatory, acutely inflammatory protein involved in inflammation regulation, like we cut our skin and get infection, and AGP2 might be released from local cells and just mediate inflammation. And this bumper molecule just mediates the function of BMP4. BMP4 is a molecule regulate formation of bone or heart. And chemokine-13, it's a cytokine. It regulates inflammation, inflammatory responses. FGF21 regulates glucose level. Just behaves like insulin, but it's not insulin. Can lower blood glucose. Actually works for mice, rats, and cats, and humans. Currently, FGF21 has been used to lower human glucose level for diabetes. NRG4 is a factor regulating neuronal growth and development. PRG4 is also a serum blood-borne protein regulates inflammation. SAA1 serum amyloid is also an acute inflammatory factor. The function has not been identified. But what we know, amyloid A1, A2 are bad. Just that they accumulate in organs and form amyloid. Amyloid clusters and conjugates, aggregates. And they just affect the normal function of the cells. So TFF3 was originally identified from intestine and the stomach. The function of TFF3 is to produce a layer, protective layer for intestine and reduce the injury, protect the intestine from injury. So now we found that all of these molecules are upregulated in the liver in response to myocardial infarction. And so the hypothesis from that was, well, these molecules might include cardioprotective factors. In the following, I'm going to show you how to find cardioprotective factors from this population of secreted proteins upregulated in the liver. So the screening method was injection of recombinant protein. Each of the recombinant proteins for the 9, well, actually we tested 7 proteins because amyloid A1, A2 are bad. We did not test those. The hypothesis was, well, these two might not do anything good for the heart, can only be bad. So we tested 7. We inject a recombinant protein for a particular liver-derived secreted protein into the circulation immediately after myocardial infarction was induced in the mouse. So this column shows left ventricular specimen slices. And this is a mouse heart. Right here, there's a white dot here. That's this location where coronary artery ligation was. And below that ligation point, from each heart, got 3 pieces, one immediately below the location, second piece from the middle here, third piece from the apex area. So each circle here is a slice from left ventricle. So the color codes. Red color shows good part, well, intact, healthy myocardium. White color is bad, infarct. So PBS means PBS, phosphate buffer saline injection, that's controlled. Induce myocardial infarction, inject PBS. Then, well, at 24 hours post myocardial infarction, you just collect these specimens. So you get that much of white color bad tissue. So bad area. And this is the CXCL13. You will get a higher, well, about the same level of myocardial infarction, bad tissue as PBS. PRG4 did not do anything good. But the remaining 5 factors, when each is injected into the mass circulation, you can see a significant reduction in the white bad area. So that's the message from this slide. So all 5 columns shows each column represents the result from after, say, AGP injection or bumper injection and so on. So the last column shows results from an animal with injection of a combination of all 5 protective factors. You can see even a more reduction in the bad area, infarction. So this is just the analysis for the reduction, for the protective effect. In short, 5 proteins each induced a significant reduction in myocardial infarction, so that therefore that protein was selected as a protective factor for the heart. Next thing we did was tested the protein level, protein expression of the 5 protective factors in the liver. So this column shows the immunoblood analysis for all 5 factors, including beta-actin, which was used as a control, loading control, just showing equal amount of protein was loaded. You can see probably there's a slight increase in some of the protein, like bumper here and FGF21 during the course post-myocardial infarction. But in myocardial infarction, you can see that at half day 1, 3, 5 days, you can see upregulation of the protein, the 5 protein in the liver in response to myocardial infarction. One point I like to point out is that a unique expression pattern for TFF3. TFF3 was higher at day 3, and then dropped a bit at day 5, and come back, get higher, much higher at day 10 and 20. So there are 2 peaks for that. Our guess was the first peak for TFF3 was early protection for the heart, and for the later peak was for some myocardial regeneration responses. But we have not looked into that issue. This simply shows the serum or blood level of these 5 proteins by using ELISA. So the message here is for all the 5 cardioprotective proteins, you can see a transient increase in the serum level from day 1 to about day 10. Some from day 1 to day 10, some from day 1 to day 5. Like FGF21 was day 1 from 5. You can see a peak increase in FGF21 in serum. Actually, in human patients with myocardial infarction, you can also see FGF21 increase. But the FGF21 function or protective function has not been found in humans. Next thing, what we did was, well, we like to know the long-term effect of these cardioprotective factors. So what we showed, we have seen was just a short-term, 24 hours protection. That was just for screening purposes. So now we're going to test what about after, say, 20, 30 days post myocardial infarction? Will the liver secreted proteins protect the heart? But one thing we like to do is in myocardial infarction, you have to detect if the protein you deliver can get to the heart. You say to cure a disease or protect injury, cardiac injury, cardiomyocytes from injury. So you have to know if this protein is present in the injury site. To test that, we used a fluorescein conjugated FGF21 molecule. And we get the protein, get fluorescein, and mix up and use some chemical assay to link the fluorescein fluorophore to the protein, and inject the fluorescein protein to circulation. And after 1 day, and remove a piece of a left ventricle here. This is left ventricle specimens. We have seen that labeled by PTC reagent. So red again is a good intact myocardium, white is infarct. You can see that the fluorescein FGF21 only ended up in the lesion, with broken heart, but did not go through the capillary network or go through capillary endothelial cells in the intact myocardium. So this is because in injury area, the permeability of endothelial cells increased, allow protein to diffuse from blood compartment into the interstitial compartment. This shows simply that any injected protein can end up in the lesion. Next, we did was the long-term protective effect of the injection of the 5 liver-derived proteins at 5, 10, 30 days. So the assay here, the assay was we used was orange, red, and brilliant blue, 2 color. The red color, orange, red, dye stains, intact, healthy, good myocardium, and brilliant blue stains collagen, simply stains fibrosis. So the left column shows PBS control. So each circle was from the heart. This was from the middle of the left ventricle. This part here, the thin layer, is the right ventricle. This circular part is the left ventricle. You can see that at 5 days, you get that much of scar, fibrosis, scar tissue, the green color. 10 days, the blue color shows fibrosis with scar. 30 days. Well, after you inject a combination of 5 liver-derived protective proteins, I can see that the blue-colored fibrotic region was significantly reduced. So this is just analysis showing you statistical significant reduction in myocardial infarction. So the conclusion was that we can use a combination of the 5 liver-derived secreted proteins to reduce myocardial infarction. So this has implication for clinical application. So currently we're developing some of the liver-derived proteins for human applications. One was TFF3, because TFF3 is the most effective single factor. And also, TFF3 protects ischemic left ventricle from rupture. For example, you use TFF3 knockout mice. You induce myocardial infarction. Within 5 days, post-myocardial infarction, about half or even more of the animals will experience sudden left ventricular rupture. The animals die within 5 minutes because the heart is broken, ruptured, because of the infarction. So if TFF3 is not there, knocked out, no TFF3 TFF3 expressed in this animal, and the left ventricle cannot just hold the pressure, arterial left ventricular pressure. But in wild type animal, TFF3 is there, and the rupture rate for the left ventricle post myocardial infarction dropped below 10%. You see there's a huge difference. So that simply means that TFF3 protects the left ventricle from rupture. So right now we're trying to understand the mechanism behind that. Could be anything inside the cell, or it could be something extracellular matrix. So TFF3 will promote the mechanical strength of the broken heart. Next thing we did was echocardiography to test the contractility of the heart, but because of time, I would like to skip this. Also, we tested the left ventricular DPDT. It's a parameter used to test the left ventricular contractility. So we'll skip this also because of time. Another thing I'd like to talk about is the action of the naturally expressed liver-derived secreted proteins in animals, in the mouse. So the question is, do these liver proteins protect heart in wild-type animals? So a common approach to test such a problem is a loss of function test, meaning that you have to remove, if you like to test the function of a factor or protein, you have to remove that factor or that protein. That's called loss of function. So here we have 5 cardioprotect proteins here. We have to remove 5. Well, gene knockout is a good model, but knocking out 5, it's very difficult. I haven't heard anyone, any lab can do that. Probably most we can knock out, it's just 2 double knockout would be very challenging. But here 5 is very difficult. We didn't even try that. So how you knock out or remove partially or completely of these 5 factors. So we took approach siRNA, small interfering RNA approach. So you simply dump a combination of 5 siRNA reagents into the circulation. And so the animals, probably including humans, have a feature. That is, any siRNA you inject into circulation, the liver will take up at least 80% of them after a few minutes. That's great, because we try to knock down or remove liver proteins, and the liver cells will take up most of the siRNA molecules. So siRNA will get to the liver and actually significantly knock down the expression of all the 5 protein genes. Look at here, say this is AGP2. PBS injection, you have that much expression in AGP2 at, say, 3 days after siRNA injection, but 1 day after myocardial infarction. So we injected the siRNA 3 days prior to the induction of myocardial infarction. And then you assay the protein level. So they used a control siRNA, meaning that siRNA does not reduce messenger RNA level for any known genes. And the specific siRNA is just the AGP2 specific. So siRNA is very specific. Each siRNA can only knock down the expression of messenger RNA, target messenger RNA. You can say siRNA treatment probably knocked down about 80% of the protein level. That really worked. Next thing we did was, well, what happened after siRNA-mediated knockdown? If you can still see, well, you can see protective factor, protective effect, where you increase or decrease cardiac protection. It turns out that siRNA knockdown induced a re-increase in myocardial infarction. meaning that if you remove a protective factor, so the heart is no longer protected by that factor. So therefore, myocardial injury or infarction will increase. So these images were from left ventricle, broken left ventricles assayed by TTC reagent. So the message here from this slide was that knockdown where you partially remove these 5 proteins from the system, from the animal, and the myocardial infarction level will increase. That will prove, partially at least, that these liver-derived 5 secreted proteins do protect heart from injury or death. So we also tested the left ventricular DPDT and echocardiography. We'll just skip that. This is for siRNA treatment. Well, in the remaining couple minutes, I will just briefly mention about the mechanism, how these liver-derived proteins act to protect the heart. I mean, you just got a bunch of small molecules, so these molecules can get to the broken heart, then how they work. So this shows the pathway, major molecules that really the liver protein get to the inside of the heart, ischemic cardiomyocytes, to eventually promote cardiomyocyte survival. For example, FGF21 first will act on FGF receptor 1. Actually, there are 4 types of FGF21 receptors, and cardiomyocytes in the mouse express receptor 1 and 3, like shown here. So here is receptor 1, cardiomyocytes from sham-controlled animals and also from animals with myocardial infarction, 1, 3, 5, 10 days. Receptor 3 is also expressed in cardiomyocytes in the mouse, but 2 and 4 are not expressed, just nature. So FGF21 turns out to act on receptor 1. The receptor 1 activates a intracellular molecule called PI3K. It's just a kinase. K stands for kinase that acts to add a phosphate group to a phospholipid molecule in cell membrane. And PI3K acts on a phosphate lipid molecule called PIP2 and turns that into PIP3, meaning one more phosphate group added to this phospholipid group molecule. And PIP3 will activate another molecule called AKT1. It's also a kinase. So a kinase will act on downstream molecule called BAT. That's BCL2 associated death promoter. It's, well, name doesn't mean anything, but that thing causes cell death. But if AKT1 is activated, AKT1 induces phosphorylation, adding a phosphate group to BAT molecule. That will shut down the activity of BAT, will reduce cell death. So that's the mechanism. So in the end, BAT is phosphorylated with a phosphate group, and it can no longer work. Therefore, can no longer induce cell death under ischemic condition. Therefore, ischemic cardiomyocytes will survive. So that's how FGF21 works. At this time, we only worked on FGF21 and TFF3. We don't know how the remaining 3 factors work. So the take-home message is that whatever the molecule is protected and cannot save dead cardiomyocytes. In a heart, for a broken heart here, you see the white area. In the core area, the center of the white area. Well, that contains dead cells. I mean, Nothing can save those cells. God cannot save those cells because they are dead for good. And the protective factors only act on the peripheral area here. Act on injured cardiomyocytes, but not yet dead. So if this signaling pathway is activated, so the injured, not yet dead cells can be saved, can be rescued. So that's the idea. Well, with that, I'd like to get a conclusion part. In conclusion, well, many conclusions we can make based on the information we just talked. But I'd just like to see one thing. That is, the liver has been evolved as an organ contributing to myocardial protection. There are two ways for the protective action. One is to mobilize cells, home cells to the broken heart, and the broken heart liver cells will release, upregulate and release secretory proteins. So these proteins will rescue injured cardiomyocytes from death. Well, I'd like thank you for being here, and I'll stop here and be glad to answer some questions.
So good. Questions?
I was wondering if you looked specifically at what percentage of the protection comes from those proteins being secreted in the blood by the liver itself, or like what role the mobilization and homing plays in that. Like if you're trying to block the homing specifically to see if the cell deals to protect, or if the factors in the blood is enough.
Yeah, that's really a question. Got to the point. We spent 2 years, we could not answer that question. Both liver cell homing to the heart and direct liver cell secretion of these proteins will work in terms of the myocardial protection. But which one is more important, I don't know. We have to, as you just said, we have to block one and look at the other. But the problem for that question is we just don't have a method to block either of these two events. Would be really nice if you can help me to to find a method to block liver cell mobilization. That would be great. We just cannot find any way to block liver cell mobilization. It's there, it's nature. I mean, you have a broken heart there. You know, heart is short of protective mechanisms. Just like brain. Any of these important vital organs, they have very limited capability or capacity of protection. We just don't understand why. And nature select the liver or some other organs. Other organs might also be involved. We just did not get a chance to look at. And nature select the liver to be responsible for myocardial protection if the heart is broken. Now you try to block that, it just cannot block liver cell mobilization. You can not block liver cell secretion of this protein either. You can use, say, well, you can use siRNA, we use that, but you cannot completely block. siRNA can only knock down about 80%. There's still 20% protein up there. This 20% protein might also play a major role. And well, for liver cell mobilization, well, you say, well, that's a physical event. You just can't do something physically to block it. We can't just not, because anything you're sticking into circulation will cause thrombosis, and the blood vessel will be completely occluded within hours or days. You say, well, why don't you just remove the liver? Well, you just cannot. The animal will die within a day if you remove the liver.
Kind of related to some liver cell issues following myocardial?
No, liver cells will not home to any healthy tissue. I don't know why. Because to get to any peripheral organ, liver cell must go through our arterial system, while liver cell will be sheared to pieces. That we have tested. Because when you can collect liver cells by using collagenase trade mount, and you drop that into a flow chamber system, and you just gradually increase the shear flow rate, shear stress, up to a critical level, and all liver cells just sheared into pieces. About the slightly below the arterial shear stress level, liver cells just cannot survive.
Oh, thank you very, very much for a fascinating talk.
Sure.
It was really great.
Thank you.